Hey, welcome back to BioinfoBytes. Today, we are going to decode the world of protein databases. We're going to zero in on three of the biggest names you'll run into: UniProt, SwissProt, and TrEMBL.
So the genomics revolution is absolutely flooding us with millions of new protein sequences every single year. But here's the thing, not all of that data is created equal. So the big question is, how do we tell the difference between reliable, verified info and all the raw, unreviewed predictions?
And just to give you a sense of the scale here, we are talking about over 230 million protein entries that are just computationally predicted, mostly from automatically translating DNA sequences. It's an absolutely staggering amount of data to sift through.
Now, compare that massive number to this one, only about 570,000 sequences. These are the ones that have been painstakingly, manually reviewed by expert scientists, and they're backed up by solid experimental evidence. And that right there is the central challenge, finding those islands of quality in an ocean of quantity.
So what's the solution that was built to manage this exact problem? Well, that would be the UniProt's database.
Okay, let's dive in. UniProt, which is short for Universal Protein Resource, is pretty much the world's most complete catalog for protein information. And it's not run by just one group. It's this huge international effort between some of the absolute top bioinformatics institutes on the planet.
The best way to think about UniProt is like a giant umbrella. Underneath it, you've got the core, which is called the UniProt Knowledge Base, or UniProtKB. And this is where it gets really smart. It's divided into two main sections: the reviewed stuff, called SwissProt, and the unreviewed stuff, called TrEMBL.
So let's start with SwissProt, which is all about one thing: curated quality.
Every single entry in here is manually checked and annotated by expert biologists. They're literally digging through scientific papers to add functional details, links to diseases, and all this other critical data. Because of this meticulous process, there's very little redundancy, and it's considered the absolute gold standard for protein information.
Now, on the complete other side of the coin, you have TrEMBL. And TrEMBL is all about automated quantity.
TrEMBL actually stands for Translated EMBL. It's this enormous collection of protein sequences that are generated automatically. Basically, they take the DNA sequences from one of the world's biggest DNA databases and just translate them into protein sequences. And because this whole process is automated, TrEMBL is just gigantic, and it grows at an unbelievable speed. You can think of it as a huge staging area for new data. The cool part is, important or well-studied sequences in TrEMBL can eventually be manually curated and get that coveted promotion into SwissProt.
So here's the core trade-off, really. With SwissProt, you're getting manual curation and super high reliability. With TrEMBL, you get automated processing but way more comprehensive coverage. The key is knowing which tool is the right one for your specific research question.
Okay, so what does one of these database entries actually look like when you pull it up?
When you look at an entry, you'll always see a few key fields. ID is its unique name. OS tells you the organism it came from. CC is where you'll find comments about the protein's function, and SQ gives you the actual amino acid sequence.
But an entry is so much more than that. It's really a hub of information. You'll also find the gene name, where it lives in the cell, and any known links to diseases. And this is huge. It connects out to other vital resources, like the PDB for 3D structure data, or KEG for info on metabolic pathways.
Now, to get the full picture, you should know that SwissProt and TrEMBL are just two parts of the wider UniProt ecosystem.
You've also got UniRef, which cleverly groups similar sequences together to cut down on redundancy and make searches way faster. And then there's UniParc, which is this massive archive of pretty much every protein sequence ever found. For most of your work, you'll probably stick to UniProtKB, but it's good to know these other tools are out there.
So, if you remember one thing, make it this: SwissProt equals quality, TrEMBL equals quantity, and UniProt is the amazing framework that brings them both together. And that is your bite-sized breakdown for today. So, now that we know where to find a protein sequence, how do we find out what it actually looks like in 3D? We're going to cover exactly that next time as we explore the world of protein structure databases, including PDB, Cath, and Scope. Make sure to subscribe, and we'll see you on BioinfoBytes.